So I'm going to present our work in nucleic acid binding prediction. I improved protein nucleic acid binding prediction by leveraging language models. So this is a sort of a continuation of what we have been discussing in the previous talk. So this is a joint work with my students, Rahmatullah Rochi, Bernard Mossad, Hussein Shubho, and Shumit Tarafdar, which has just been published earlier this year. So protein nucleic acid interaction is fundamental and underpins a wide range of cellular processes, from gene replication to regulation to signal transduction to metabolism, and therefore characterizing these interactions are key and highly desirable, and particularly if we can do it in a large-scale manner, that's going to be of fundamental advance. Centrally, what we are interested in understanding this interaction in atomic detail. So we would ideally like to know the three-dimensional structure of the bound form when protein interacts with either the partner DNA or the partner RNA molecule. However, the experimental structure characterization or determination is not always feasible or practical. And it turns out that computational methods are not promising as protein-protein or protein monomer interaction in this particular domain, as seen here, where I'm just cut and pasting the extended data table 1 from the AlphaFold 3 work. And there are a lot of numbers, but what they're reporting is the prediction accuracy of different biomolecular prediction tasks for AlphaFold 3 work. And if we zoom into the nucleic acid task, in particular protein RNA interaction, we see the accuracy of AlphaFold 3, which is measured in interface LDT score, which ranges from 0 to 100, is about 40%. So we have significant room for improvement in this problem. So it turns out even though atomic level structure characterization or prediction is difficult, there is a coarser grained version of this problem, which is also the focus of this work, is the binding site prediction, where we are just interested in characterizing the binding site of protein DNA or protein RNA molecule. Here I'm showing the protein in surface representation in gray, where the binding sites are annotated in green. So ideally, we would like to know the binding site without any knowledge of the partner molecule, partner nucleic acid. And in particular, the binding sites are defined when the atom pairs, one from the protein, one from its partner nucleic acid, is within their sum of van der Waals radius plus 0.5 Angstrom. This is following the literature. And however, the experimental characterization of even the binding site turns out to be time-consuming and expensive. And therefore, there is a critical need to develop computational method to be able to predict binding site. And typically, these methods take a protein-centric view of the problem. In other words, they start from the protein structure in its unbound form and trying to characterize the binding site or predict the binding sites. So you go from the protein monomer here shown in the left, and then be able to predict the binding site without any knowledge of the partner nucleic acid. And this is why this problem is also known as a partner-independent protein nucleic acid binding site prediction problem. A number of computational methods have been developed in this area. So this is a very well-studied problem. Roughly, they can be categorized into two categories, so sequence-based approaches and structure-aware methods. So sequence-based approaches use a sequence and evolutionary information, typically in the form of multiple sequence alignment or PSSM, and then apply a large number of methods, for example, statistical approaches or machine learning methods. The advantages of these methods are obviously sequence data is readily available, but this tends to be less accurate compared to the structure-aware methods, which also leverages structural information. A number of methods such as graph bind, graph site, these used to be the state-of-the-art methods in this domain. These generally tend to be more accurate. Well, this method had one weakness in that they needed structural data, which may not be always available, but the AlphaFold database and the advances of AlphaFold 2 largely address that problem. So we can largely feed AlphaFold structures into them. However, these methods do not, at the time of publication of our work, did not take advantage of protein language model embeddings. So we actually looked into the problem of, can we improve these accuracies further using protein language model? And are there any other advantages in addition to accuracy in using protein language model embeddings? And this led to our work, which is PLM-informed equivariant deep graph learning method, which we call EquiPNAS. Here I'm showing the flow chart. This has three major parts. In part A, which is on the left, we have the input itself, which is typically the unbound protein monomer structure. We also have the sequence information and the evolutionary information in the form of MSA and PSSM. We also embed the ESM2 embedding, the PLM embedding from ESM2 model. And then we actually, in the panel B, we have the equivariant, E3 equivariant graph neural network approach, which is actually doing the prediction task. So what we do is we take the input monomer and then convert into graph, where each node in the graph is basically one residue of the protein. And we actually draw an edge between the nodes if the two residues are interacting or close in space. And then we apply E3 equivariant graph network, which basically leverages coordinates of the protein, coordinates of each residue, and then sequence-based and structural-based node features, as well as edge features, to do graph convolution, and then to eventually perform graph node classification for residue-level binding site prediction. So ultimately, what we will be able to predict is for every residue in the input monomer, a likelihood values which ranges from 0 to 1, where higher value represents more likely to be a binding site. And then we can apply a threshold to actually convert into a binary prediction task. So this equivariant graph network that we used had 12 layers with hidden dimension of 768 and dropout rate of 0.1, which are determined through cross-validation in a data set. For features, we use pretty standard feature set, amino acid, PSSM, MSA, but we also added the PLM embeddings from ESM2. Similarly, for structure-based feature, we use secondary structure, solvent accessibility, and geometric feature, pretty standard features. Because we use equivariant network, we also use coordinate feature, which are basically this carbon alpha coordinates of the input protein monomer. For edge features, we use a ratio of logarithm of the sequence separation of the interacting residue pairs and their Euclidean distance. And the intention here is to actually give more emphasis to the long-range interactions, which are actually close spatially. For training our method, we actually use pretty standard, widely available data set from the BioLeap database. So there are two separate databases, one for the protein DNA task, another for protein RNA task. And these databases are pre-processed to filter out protein chain with more than 30% sequence similarity between the train and test set to prevent homologous information from leaking from training to test using CD hit. I should also note that this database is the data sets themselves are highly imbalanced. You can see the number of binding residues are far less compared to the non-binding residues. And that is to be expected, but as we shall see soon in the results, this will have an effect. To evaluate our method, instead of running our method trained model using the test set only once, we actually randomly sampled 70% of the targets from the test set and repeated that experiment 10 times and recorded the average. So here we are showing the average, the means and the standard deviations of our method compared to the two state-of-the-art structure-aware method, graph bind and graph site, for the protein DNA binding prediction problem. So a couple of things to be noted. First is the area under the ROC curve is actually very good for all methods, but the PRAUC, area under the PRAUC curve, is not so good. And this is because of the class imbalance problem that we face in the data sets. We also computed the p-value of the performance difference from our method compared to the others. We found our method to be statistically significantly superior or better than the others. And same results can be found in protein RNA binding prediction task. I should mention that this is a different model trained differently for protein, specifically for protein RNA binding prediction. The performance difference here is a little more noticeable compared to the protein DNA binding prediction, but nonetheless, the problem of data imbalance still plagues these results as well. A couple of case studies to illustrate the advantage. So here I'm showing on the left protein DNA prediction, on the right protein RNA prediction for two proteins or two complexes, protein nucleic acid complex, where I'm characterizing the predicted interface or predicted binding site on the bound form, where the greens represent the true positive interactions predicted, the red is the false positives, and yellow is the false negative. We can see that our method actually turns out to be reasonably well in protein DNA, and although it predicted some red and false positive interactions in protein RNA prediction task. But the question is where this performance is coming from. Our main contribution, as I mentioned, is embedding of the PLM or the ESM2 PLM embeddings. So what's their contribution? To investigate it, we did some ablation study using 5-fold cross-validation, where we systematically removed the PLM embeddings and the evolutionary information from the model and recorded the performance. It's interesting to note for both protein DNA and protein RNA prediction tasks, when we remove the PLM embeddings, there is a noticeable performance drop compared to the full-fledged version. However, when we actually remove the MSA or PSSM, the performance drop is not as much, which suggests that the PLM embeddings contribute significantly for our performance, and that's a major driver. And we took a model, the ablated version, which does not use PSSM or MSA, and then apply it to the test set. We saw that the performance, while it's lower than the full-fledged version, is still better than the then state-of-the-art model, which means that PLM embeddings, which suggest that PLM embeddings may be able to help reduce the dependence on the availability of explicit evolutionary information without necessarily a dramatic drop in accuracy. Is there any other advantage of this? Well, obviously, if we can bypass the search for explicit evolutionary information, the MSA particularly, then there will be a speed up. And so we compare the ablated version that does not search MSA and with the version, the full-fledged version, and we see orders of magnitude speed up. For example, the blue dots are the ones that do search the MSA and PSSM. The red ones are the ablated version, which is just PLM. And so we see orders of magnitude speed up in that regard, which means we can actually do a large-scale study using PLM-only embeddings using our method. We also, because we use equivariant neural network, we wanted to understand, is there any need or necessity for using equivariance? And to understand that, we did a couple of experiments. One, we actually compared the equivariant network with the invariant counterpart on everything else being equal, same training data, same hyperparameter, same test set. And then we found out that equivariance almost always helps to get better performance. We also actually tried to use as input experimental structure instead of AlphaFold predicted structure. All the results I have presented so far use AlphaFold 2 structures as input. So we wanted to understand, what if we switched from AlphaFold prediction to native? Are there any gain? So that we noticed a slight accuracy gain when we switch from AlphaFold 2 to experimental structure, but AlphaFold 2 is pretty comparable in terms of accuracy. So main conclusion here is equivariance helps, and if we have native structure, probably we should use it. But this is our computational method, but we wanted to actually experimentally validate our method. For this, we have collaborated with a biochemist who actually came up or pioneered this method called GECX RNA. This is an in vivo method for genetically characterizing, or I should read from here. So it's called genetically encoded chemical cross-linking of protein with target RNAs in vivo. This was published in Nature Chemistry last year. So what it does, it induces or incorporates site-specific unnatural amino acids that can cross-link with RNAs in close proximity of the RNA binding proteins. So we actually took this system and applied to CRISPR-Cas system. In particular, we looked at the PSB Cas13b protein, for which structural information is not known. And what we did is we took the unbound form of PSB Cas13b protein and then used our algorithm to predict the binding sites using the protein RNA trained model. And then it turns out the residue 380 in that structure, which is an arginine, turned out to have the highest predicted probability. So we asked the experimental collaborator to actually mutate that to convert that into unnatural amino acid. And then they run the modified unnatural amino acid and incubate it with the CRISPR RNA. And then in gel running experiments, it turns out this sample, which has unnatural amino acid, showed slower running band, which means it was cross-linked. So which is a kind of a preliminary evidence that we can actually, the experimental validation that we can actually verify this prediction using experimental validation. I should also mention that GECX RNA is a high throughput method, meaning that we can now scale this up proteome-wide, which is what we are now conducting. So we are now leveraging our trained model to actually do proteome-wide validation of our computational prediction. So with that, I'd like to summarize the talk, reminding the contributions and the future work. So the major contribution is to integrate PLM embeddings for improved protein nucleic acid binding site prediction. We looked at both protein DNA and protein RNA binding site prediction tasks. And we found that PLM embeddings, in part at least, bypass the need for explicit evolutionary search, which leads to speed up and perhaps scalability. We also made our code freely available in GitHub, including not just the inference code and the model weights and the data, but also the training code. So if there is a new database that you would like to train our models in, please feel free. In terms of future work, so I should mention one limitation of our work is just looks at like two-body prediction. There is one protein monomer interacts with one RNA or DNA. But in reality, we should look at higher order oligomers beyond these two-body interactions, which is one thing we are working on. The other thing, obviously, is I think this model could benefit from better embeddings from language models, in particular ESM3 embedding. So it could be interesting to actually see the effect of ESM3 embedding. I would like to, in closing, thank the PhD student, my collaborator, the public databases, Protein Data Bank and BioLip, without which this work would have been impossible, ISMB and 3D-Sig organizers, and funding sources. Thank you very much for your attention.
Thank you very much.
Hi, Javier here. Very nice work. I was wondering, I work with ligand binding site prediction, and I believe in that context, evaluation at the pocket level or the site level is more meaningful and more informative than at the residue level. I was wondering if in nucleic acid protein binding site prediction, if you look at all at just how many sites do you recall, and not so much the residues that you predict as binding or non-binding.
We did some of that study. This study, I should mention, is a follow-up to a previous study that we did for protein-protein interaction, in which we looked at some of that. So I guess we could certainly look into that.
Thank you. Lovely talk. Loved the validation. So 2 questions. One, I noticed you used ESM 15 billion.
Yes.
So there is some sort of debate in the field as to how big the model needs to be. So I wonder if you did 650 million and 3 billion and how those did.
Great question. In the published paper, actually, we compared our model using different embeddings from ESM, and we found that there is not much of a significant advantage using the big models. So 650 is about the best we can do. So all results are presented using that model.
Okay. Second question, just quickly. Did you consider just getting rid of all the equivariants and using fold-seq embeddings? And if that makes things simpler.
That's a good suggestion. We should probably do that. Okay, no, thanks. We have not. Thank you.